Good evening, everyone. As we know that medicinal chemistry is always geared towards the drug discovery and development. The focus on development of new synthetic drug compounds that has resulted in the incorporation of many other disciplines such as biochemistry, molecular biology, and the medicinal chemistry. But when this medicinal chemistry is turned towards generating the potent biological activities in terms of generating the heterocyclic organic compound, so if you want to know about the same, you have to stay tuned with us throughout this lecture. Hello everyone, once again this is Karishma Sharma, the host of Bio Skill and Analytics Development Forum, and I welcome all of you in our today 20 session. It is 2020 session today. So thank you for joining, and it's time to introduce the guest of the day who will be going to give you information about all the concept of medicinal chemistry and will tell you about the heterocyclic compound. So it's my pleasure to introduce Dr. Prashant Rai. Thank you, Prashant, for doing this for us. Can you hear us?
Yeah. Am I audible?
Yeah, you're absolutely audible. Very good evening, and thank you for joining today in our session. We are privileged to have you.
Yeah. Thank you so much. Yeah.
I would like to tell to my all the audience. Well, Dr. Prashant Rai has hold a PhD degree in medicinal chemistry from Sri Sathya Sai Institute of Higher Learning in Andhra Pradesh. He is currently working as a research executive at Karkinos Healthcare, a cancer care platform in Mumbai. He was also a visiting scientist at IIT Gandhinagar, and his expertise lies in designing and synthesizing, as I said, synthesizing the heterocyclic organic compounds with potent biological activity. And apart from the same, he's a wonderful writer and published his 4 peer-reviewed journals along with the talks in national and international conference. As he's a scientist and also a student earlier in his life, he's also having the we all are having the activities, and he's having an interest towards the guitar, sports, and avid reader. So that's our privilege to have you today with us. So Dr. Prashant, you can start with the same.
Good. Yeah. Thank you so much for the kind introduction. And at the very outset, I'd like to thank the organizers for having me here today. I'll quickly share my slide. Just a second. Is my slide visible?
Yes, it's absolutely visible.
Okay, yeah. A very good evening to one and all. And I welcome you to another session of this journal club, wherein today I'll be talking to you about an introduction to drug discovery and development, which is specifically a journey of a drug molecule from lab to the market. To give you a disclaimer upfront, it's very arduous journey, very tedious, time and effort demanding endeavor, and with a very high risk of failure. The drug discovery and development, if you look at the stats, 99% of the time, the scientists end up not getting anything, not getting any result. So if you would ask me, why would researchers take up such an endeavor, which is very risky, which is so prone to failure? And I think I would be able to explain that answer in the subsequent slides. We all believe that the global chart looks like this, right? But what if I tell you that actual landscape looks like this? You see the entire world, each and every part of the world at some point or the other have been affected by various diseases, be it Ebola, be it Zika virus, be it cholera, be it malaria, be it COVID. Each of these parts of the world have been affected by these diseases, and these diseases have claimed lives of millions of people at various time points. So this I think gives us an idea about why drug discovery and development is important, because drug discovery and development provides an answer to, if not almost, most of these diseases. To give you the brief overview of drug discovery and development, this involves discovery phases, preclinical studies, clinical trials, and review and approval. The discovery phase itself spans over 3 to 6 years, and then when you move on to preclinical studies, wherein the compounds is tested against the animal model, After that, we move on to the clinical trials wherein the compound is screened against human beings. And finally, if everything goes right, the FDA reviews and then finally launches the drug in the market. In the discovery phase, the first step is target identification. Then we go on to target validation, lead identification, and lead optimization. The details of each of this step I'll be talking to you in the subsequent slides. When you move on to the preclinical studies, it's more about identifying and establishing pharmacokinetics and pharmacodynamic studies, especially in the animal models. We profile the toxicity levels of each of these drugs. And then we move on to the clinical trials, wherein these drugs are tested against human beings, which is conducted over three phases, phase one, phase two, and phase three, wherein we specifically look at, the dosage, safety, and the off-target effects of these drugs. And upon successful completion of these clinical trials, if the reports are satisfactory, the FDA approves the drug and launches it in the market. But once the drug is launched, the constant monitoring happens for any off-target effects. And if there's any report of off-target effect in any of the patients, the drug is retrieved back from the market. Starting with the discovery phase. My slide is not moving. Yeah. The first step in drug discovery is target identification. So what do I mean by target? Target here refers to a drug target, which could be protein or genes, which is responsible for the progression of the disease. The first thing to look out when we do target identification, some problem with my slides, just a second, I'm not able to move it. Just a second, I'm not to a slide. Yeah. Yeah. So the first step involved is literature review of the drug. So yeah, in literature review, what we do is we understand the types of diseases. We understand what are the related pathways involved in the disease. We also study what are the symptoms that the patient is showing, and then accordingly try to decipher what could be the way out further. Then we go on to the genomics and proteomics studies. In genomic study, we do gene expression profiling. So basically, we look at the gene profiles of the healthy tissue or cells and compare it with the diseased one. And then we look at various gene expression and see which gene is overexpressed or which gene is not expressed at all. So this way this enables us to identify the genes based on which we can say that this gene is responsible for the progression of the disease and could be the potential target. When it comes to proteomics, it deals more about the protein landscape of the biological system. And in proteomics, similarly, we look at the protein profiling of the healthy cells compared to that of the diseased ones. And then, we identify which proteins are expressed in the diseased ones and see that if that protein could be targeted. The computational studies involving molecular MD simulation, that is molecular dynamic simulations and molecular docking, helps us to identify what are the probable sites of attack, which means the active sites or the allosteric sites of the protein that could be targeted by the drug. It also tells us about the structure of the protein and the possible conformation, the thermodynamics involved in the binding between the drug and the protein, and gives us an idea about which could be the best probable target for our drug molecule to go and attack. The fourth step is the biomarker identification, which is an indication or which is an indicative measure of the presence of disease condition. Biomarker confirms the presence of disease, and we all know about prostate-specific antigen, which detects prostate cancer, or HER2, which predicts breast cancer. Biomarker identification also helps us in stratification of the patients and enables us to find out which group of patients could be more responsive to the particular treatment. Next step after the target identification is the validation of the target. This is basically to ascertain whether the target that we have identified is actually responsible for the progression of the disease. So for target validation, we carry out transgenic knockouts. So transgenic knockouts basically involves transgene, which is an exogenous gene, which is incorporated or introduced into the animal model. And then these genetically modified organisms are studied for the progression of the disease. So a particular specific genes are completely knocked off, and then we see whether the disease progression happens. And if not, then we are sure that that particular gene is responsible for the progression of the disease. The genetic tool like CRISPR-Cas9 or TALEN helps us to carry out this transgenic knockouts kind of study. The second method of validating the target is mRNA modulation. We all know that mRNA is responsible for the transcription and eventually the translation of the protein. And hence by altering the levels of mRNA or by doing the analysis of mRNA, we can make it bind to siRNA, small interfering RNA, and then block the translation of certain proteins. So we could then understand whether that particular protein is actually responsible for the progression of the disease or no. The third method of validation is biomarker validation, which involves a kind of quick clinical study wherein we take the biological samples from patients and healthy controls and try to see whether the level of biomarker is actually indicative of the disease condition. We do the case control studies wherein we follow up with one patient and over the period of time monitor the case, or we take the prospective cohort studies wherein there are a vast number of patients and we try to correlate and compare it with the healthy volunteers. After the target identification, the validation, we then move on to the most important uh, step, which is organic synthesis. So organic synthesis involves designing of the compounds. Designing of the compounds includes, coming up with the compounds, its reaction pathways, its methodologies, and then finally synthesizing it in the lab. So you synthesize the design compounds. You do purification by HPLC, flash chromatography, and then you validate the structures of the compound using spectroscopic methods like nuclear magnetic resonance, NMR, LCMS, FTIR, and UMS. All these methods. So once we validate the compound, then we do in vitro screening against cell lines so that we get to see what are the therapeutic effect of the design compounds. I just want you to look at the number here, number of compounds. I have shown it to be 1,000, but I am being very optimistic over here. The library of the compound generally starts with 10,000 to 15,000. And based on its therapeutic effect, when it reaches the preclinical trial, the number comes down to 150. Further on, because of the pharmacodynamics and pharmacokinetics property, because of the toxicity profiling, when it goes to the research, clinical research, the number goes down to 10. And finally, out of 10,000 to 20,000 compounds, we'll end up with one approved drug. As we talked about screening, so what are the ways in which screening happens? The screening happens in 3 different ways, which is phenotypic screening, target-based screening, and fragment-based screening. In phenotypic screening, we screen the compounds and drugs against the complex systems, which are 2D cell cultures or organoids, which in case of cancer mimics the actual tumor in the biological system. Here in phenotypic screening, it's not necessary that you should identify the target. You can just look at the phenotypic effect of the desired compounds, and you can also ascertain the toxicity of the compound in normal cells. When it comes to target-based screening, it's a little more direct because here you leave out all the environmental factors and just see the binding of your compound with a particular target. For example, kinase. So in this case, you just take biochemical assays like kinase assays, and then you see whether your compound is particularly binding to that particular enzyme. The third mechanism or third method of hit identification is fragment-based screening. So in fragment-based screening, you don't take the entire molecule, you just take a small fragment of it. And you do that because you would provide a bigger chemical space. If you find out that a small fragment or a scaffold is very active against particular disease, you can then have n number of options to modify it with the subsequents, keeping the backbone same. So it gives you the more wider chemical space to provide you with the synthetic options. So how do we do fragment-based screening? We take the fragment and take the target protein and do the screening. Here we use more advanced biophysical techniques like nuclear magnetic resonance, X-ray crystallography, and surface plasmon resonance. So how do we do in each of these techniques? I can give you a brief description of it. In NMR, you take the protein, you take the fragment, and then you record the NMR spectra. And then from the individual spectra, you'll be able to compare and see whether the interaction is happening or not, because there'll be the definite shift in the chemical, in the protons or carbons involved in it. In X-ray crystallography, you do co-crystallization of the fragments along with the protein, and you do X-ray crystallography and get a clear image of crystal image of the fragments and the protein bound together. In surface plasmon resonance, you take the protein on the chip and then you incorporate the fragment along with it, and then there will be the change in the resonance angle when these protein and fragment binds with each other. And based on the resonance angle, you'll be able to real time monitor the interaction between the fragment and the protein. So once you screen the compounds, once you identify the hit compounds and the lead compounds, you do further optimizations of the compounds. This is basically done for improved pharmacokinetics and potency for improved efficacy. So one of the main reasons, reason why we do lead optimization is it gives us an opportunity to establish structure activity relationship. We'll be able to observe and identify certain chemical structures or backbones which might be more potent, and we can use that knowledge of that structure and activity relationship between the scaffolds and the target protein, that will be able to incorporate substituents or chemical groups which are more reactive, and hence will be able to modulate the activity of the hit compounds. We do the iterative optimization, which is like repeated cycles of modification, testing, because all in during the lead optimization, when we are doing chemical modification, we should ensure that we are not losing out on its inherent biological property. Because when you modify the compound for better efficacy, it should not happen that it compromises on your pharmacokinetics or your solubility. So you have to keep in mind when you are juggling with the structure, the basic inherent property of the hit compound remains intact. So once you do the screening of the compounds, once you've identified the hit compounds and the lead compounds, we move on to the preclinical studies, which is mainly carried out in animal models. This is mainly so this is for the first time the screening will be done in the biological system. So in this case, what happens is we basically ascertain the dosage, the toxic effect of the drug, the pharmacokinetics, which is basically ADME properties, absorption, distribution, metabolism, and excretion. And we also will be able to identify the mechanism of action in each of these cases. Basically, the drug safety in preclinical studies is very important, because after preclinical studies, we take the studies in the human beings. So the human lives, as they say, is more important. That's the reason why we make sure that the safety aspect of it is properly profiled. Pharmacokinetic studies. So as I told you, the in vivo studies in the animal model gives us an idea about the pharmacokinetics of particular drug molecule, which is absorption, distribution, metabolism, and excretion. Absorption basically gives us an idea about how the drug molecule reaches the circulatory system from the site of administration. It could be either the mouth or the GI tract, or it could be the skin. Then how the molecule reaches various tissues and organs of the body is given by the distribution. Then how each of these drug is broken down into its metabolites so that it can give the desired effect. The metabolism aspect of it profiles it. And then finally, how the body eliminates it. It either happens in kidney or liver. The excretion of it happens. So this is the ADME properties that is profiled during the in vivo studies. As I told you, the toxic effect, ascertaining the toxic effect of the compound is very important. And when it comes to toxicity, they generally profile acute toxicity, which is basically evaluating the effect of the compound at a very high dose. The chronic toxicity, wherein you have identified the right dose, but whether that right dose or low dose is causing any impact in the long term. Then there's the genotoxicity, which is ascertaining whether the drug is damaging or causing any mutations. And then the cardiovascular safety, which is basically related to the effect on circulatory system and heart. So once all these preclinical studies is carried out, we then move our study into the human beings, wherein we carry out clinical trials in three different phases: phase one, phase two, and phase three. After which there will be an FDA review and approval. So in phase one studies, in phase one trial, the study is carried out in healthy individuals. This is basically to see if the drug is safe enough to be administered. The doses have to be evaluated, the off-target effects have to be profiled so that uh, the healthy individual doesn't have any problem. Once the phase one trial is done, we then move to phase two, wherein we do the drug administration or screening in the patients suffering from the disease, and then we generally look at the efficacy evaluation of it, how potent the compound is, whether the compound is able to bring the therapeutic effect, desired therapeutic effect. Once that is done, then we then move on to the phase three trials, wherein that involve a large number of patients, and longer treatment duration. So you administer or you treat the patient with the drug molecule, and then you monitor them over the period of time, both longitudinal and also in a cohort type. So that way you'll be able to see whether there is any off-target effect, whether there is any toxicity, or whether there is any problem with the drug molecule getting into the biological system. So once phase one, phase two, and phase three um, clinical trials are successfully done, then upon satisfactory result, the FDA, that's Food and Drug Administration, reviews the result, and then finally uh, they commercialize the drug. But the constant monitoring of the drug happens, and as I told you before, if there is any off-target effect reported even after the drug is commercialized, the drug can be retracted back to the lab, and then there has to be again the optimization of it, the toxicity profiling, and all the way through. So what are the challenges in drug discovery and development? As I told you before, it's a very lengthy process. And why is it lengthy? It's because of the complex regulatory environment. There are strict rules for safety, for quality, and for the right reasons, of course. Then there's high rate of failure. High rate of failure is because the compound has to go through so many screening, and then it's very difficult when you start with 10,000 molecules and you not end up with the drug in the market. So it's a very risky endeavor that you take up. But as I told you before, uh, if you get a drug in the market, it's the breakthrough. The last aspect of it is high R&D cost. Yeah, definitely, because we see that millions of dollars are being sold out when it comes to uh, rolling a drug in the market or developing the drug. So in order to make significant financial investment, it becomes very risky. And then the pharmaceutical companies, they invest lots of money in order to roll out drug in the market. With the advent in technology, these are the emerging trends in drug discovery, wherein with artificial intelligence coming into the picture, it definitely has accelerated the growth of target identification, I would say, and also the data interpretation and giving us uh, uh, lots of insights on molecular interactions. But again, the artificial intelligence can only do so much. The entire process of synthesizing the molecule, carrying out clinical trials cannot be done by artificial intelligence. And hence, there might be a shorter time for target identification that could be carried out with the help of artificial intelligence. But when it comes to carrying out preclinical studies or hit identification, still the lab people have to work in slogging day in and day out to do that. So artificial intelligence cannot help much in that aspect. But yeah, definitely it accelerates the identification of potential target candidates by identifying the potential target and validating the target. With the genomics and biomarkers, we are able to roll out personalized medicine, which are like the treatments which is tailored to particular individual patient. So this is because they can have improved efficacy without any off-target effects. If we see all the drugs rolled out in the market that is synthetically made have
Hi,
some
sorry to
off-target.
interrupt, but
Yeah.
are you showing the emerging trends in drug discovery slide?
Yeah, is it not seen?
Okay, okay,
Which
okay.
which slide are you in?
So someone said that sorry for interruption, but slide is not visible. There might be problem
Is it visible now?
on your end. Yeah, is it visible? It is visible. Okay.
Okay. Can you just read out which slide I'm in right now so that I can be sure that you are able to see?
Emerging trends in drug discovery.
Cool. Yeah, yeah, yeah. Is the problem that's happening or it's just now it's surfaced? The
No, it's
slides.
okay. Okay.
It's okay, right?
No.
Cool. Yeah. So as I was telling that the drugs which are like the compounds which are synthesized in the labs, they always have an off-target effects. And if you see the current cancer treatment, like the compounds like cisplatin, the drugs like cisplatin or 5-fluorouracil, which are very effective, they do have an off-target effect. But it has been in use a very long period of time because there are no other drugs which are as effective as that. So people have to put up with off-target effects. So because of that, now biologics and antibody therapies are gaining more prominence because with the advent of CAR T cells or TILs, the toxicity is reduced, and you take the cells from the patient and then you culture it in the lab and then put it back there in the biological system and they help fight the disease. So CAR T cells, tumor infiltrating lymphocytes, these are monoclonal antibodies. So these are currently gaining trends, and in the coming days, it's been reported that these therapies are also pretty very effective. So this takes care of the off-target effects to some extent. And the next thing is repurposing existing drugs. So as we all know that the drug discovery and development takes a very long period of time for the drug to come out in the market. And in cases like pandemic, like COVID-19, if it hits or when it broke down, there was no drug available in the market. So and at that point of time there is no way that the new drug could be developed. So in that case, people started the scientists started looking at the existing drugs which are FDA approved and try to figure out and try to determine whether those drugs could be used for the treatment of the current disease. So if I correctly remember, hydrochloroquine and chloroquine were tried to be repurposed and reused for the treatment of COVID to some extent. So this is basically drug repurposing is basically done so that the treatments to the patients can be brought more quickly. Coming to the summary of the talk that I've given, I started with the drug discovery phase, which involved target identification, validation, hit identification, and then the optimization. Then moved on to the preclinical studies involving in vivo systems, pharmacokinetics, pharmacodynamics, and toxicity profiling. Then moved into the clinical studies wherein the testing is done against human beings. And then finally, the challenges and emerging trends in the drug discovery. So this is how I try to give the brief overview of how the drug discovery and development process takes place. I would like to tell you that each of the step itself could be a presentation in itself. I mean, if I look at drug discovery alone or look at preclinical studies alone, that is a huge topic in itself. So I just try to give you a broad eye view of how the drug discovery and development process happens, and I hope I could convey to you in simple terms as to how the process goes out and then how the drug gets discovered and developed. Thank you very much for your patient listening, and I'll be happy to take any questions from your end at this point in time. Thank you so much. Any
Participants,
questions?
please ask your questions. There is one question in the chat. Please explain
Okay, I'll
again
just
hit identification after completing the webinar.
uh just one second. Let us check. So okay, I see one question there from Avantika. Great presentation, thank you. Regarding the process of repurposing drugs, does it adhere strictly to the user hierarchy or can it be potentially skip certain step phases, resulting in the faster process? Yeah, that's a good question. The very idea of repurposing drug is to bypass all the FDA screening and clinical trials, preclinical studies, because it has already been approved by the FDA, which means that it can be safely administered in the biological system. So you don't have to go through the process of screening it, clinical trials, preclinical studies. You can directly test it in the patients, and then see if it works. That's a boon. So that is the advantage of repurposing drug. And to answer your question directly, no, it doesn't require FDA approval after it's already been rolled out in the market. I don't see any other question in the. Okay, I see one question from Ajit Pathak. Please explain again hit identification after completing the webinar. I can explain it right away. So what happens? Okay, what happens in hit identification is you are trying to synthesize the compounds. It's a synthesized library of compounds, and then once library of compounds synthesized, you test your compounds against in first in vitro in the cell lines. For example, let me consider cancer. Uh, if you're treating, if you're doing research in cancer disease, and what we try to do is we see whether our compounds are active against the cell lines, the cancer cell lines. So that's the first step of screening. So hit identification, basically hit is basically the hit compound. Like what would you say? Like is the super hit kind of the most potent ones. So yeah, it's the idea of picking out the potent molecules from all the library of the molecules that you have synthesized. So then you take up those hit compound which are very effective against the cancer cell lines. You take it up and then you screen it further in vivo models like mouse model, do optimizations of it. Suppose it might happen that your hit compound might have low solubility, so you would try to play around with the molecule. You would try to increase the efficacy by introducing covalent inhibitors or hydrogen monoacceptor donor. So that juggling you do in order to get the best activity, and in order to solve all the existing problems that's associated with the molecule. So that is what hit identification is. I hope I'm able to make sense, but if I'm not, then I can again get back to you after the webinar.